and thank you, Rakesh, for getting us started today. Good morning, everyone. I'm going to let you read for yourself my disclosures. An educated audience like this doesn't need me to read it for you. I'm going to be showing you some exemplars today that will speak to bottlenecks in developing the next generation of immunotherapies, and we'll get on to the contents now next. I'll go over drug platforms and strategies briefly to give us a little time back for Q&A. Rakesh has covered some of this himself, so where there's overlap, I'll speed through. And as I just commented, using exemplars of our early development portfolio, the OX40 and ICOS agonist programs, I will pause in giving those examples occasionally to point out very specific bottleneck challenges to be aware of for developing your own portfolio and whatever role that you serve in your organizations. I'll sum up quickly and offer acknowledgments, and then we can hopefully have some time for Q&A. I've seen some thoughtful questions starting to come in already. So this has been reviewed before in multiple reviews, thoughtful reviews from Ira Melman and colleagues at Genentech. This citation here is from a review that my lead chemist, Jerry Adams, and I and others wrote a couple of years ago. And I would say that at this point, if there's someone that isn't convinced that immunotherapy has promise for cancer, they just really haven't been paying attention in the last decade. So I know I don't have to sell that point to you. The real point I wanted to make here is that we've only really tipped into this space in terms of looking at checkpoint modulation, T-cell checkpoint modulation specifically, and blockade of exhaustion or repressive checkpoint. There's much more to do here, a plethora of opportunity, and to the points that Rakesh made about safety, we also need to bear in mind the safety parameters in developing new immunotherapies. But certainly, regardless of platform, there's a lot of opportunity in this space besides just antibodies, and that's illustrated for you here, another table from that publication from a couple of years ago. We at GSK, we've invested primarily in antibodies, small molecules, and autologous T-cell therapies. We certainly have a large vaccines team here at GSK, but not per se in oncology anymore. And this actually, just in considering platforms, in addition to what you heard from Rakesh, I would say this offers an initial bottleneck at the very earliest stages of considering how to go after a particular target axis, thinking very carefully and translationally where appropriate about what platform to pursue is paramount. In some cases, you don't have a choice, right? If the target or the target axis is largely intracellular and of an enzymatic nature, a small molecule is probably going to be the only platform of choice. But in other cases, for checkpoints and other immune axes that at least have as part of their signaling pathway an external component, a receptor ligand axis, you can approach this through at least 2 or 3 different platform strategies. And so learning from what has worked and translated in the clinic so far with existing therapies, I think is paramount for thinking about what type of approach you want to take with your new target that you're considering in immuno-oncology for a next generation therapy. And that's one of the bottlenecks that can represent either a fast forward opportunity for patient benefit, or if you choose incorrectly, you can end up in the back of the line. Even if you're first in class, you may end up in last place in terms of a patient advantage simply for making a poor choice or a non-veteran choice of platform. I think the combination value is certainly apparent. With all the trials, you saw a reference that Rakesh showed you, hundreds of combination trials with PD-1 blockade alone. Some of those starting to show some impressive benefits, such as the one that I'm citing here, but most of those still not evincing the kind of transformational combination value that we'd like to see for our patients, including the durability. And so one bottleneck that you may want to think about and looking at it preclinically before you get to the clinic is which combinations are going to be the most thoughtful, which are potentially synergistic based on the biology, based on a preclinical ex vivo or in vivo evaluation before we get to patients, so that ethically we can approach combination strategy in the clinic in a prioritized way and not really do the experiment in totality in our patients who give us, through their patient hope, the most precious contribution to our trials, and that hope is expended typically once in a clinical trial. So to be thoughtful of our patients and that value, having an understanding of which combinations show the best promise before you get to the clinic, I think is both ethically and strategically critical.